Hello everyone, my name is Nitya Banerjee. Here am I going to speak about cloning. So first of all, we need to know two important enzymes that are involved in cloning, restriction enzymes and DNA ligase. Restriction enzymes are those which are also known as restriction endonuclease. They recognize and cleave a DNA at specific nucleotide sequences, generate smaller DNA fragments, and can have either sticky or blunt ends. Sticky ends are having overlapping complementary ends. Blunt ends are not having any overlapping complementary ends. Sticky ends having that can pair among themselves also. The other enzyme is ligase. It basically joins the DNA fragments and the vectors among themselves. So there are certain types of restriction enzymes. The major, there are three types, type 1, type 2, type 3. So type 1 and type 3 has random cleavage sites which are far from the recognition site. For type 1, it is almost 1000 base pairs from the recognition site. For type 3, it is somewhere around 25 base pairs downstream of the recognition site, while type 2 is specific and the cleavage site is the same as the recognition site. There are two activities of the endonuclease and methylase are separate entities in case of type 2, while it is in a same single protein molecule in case of type 1 and type 3 restriction enzymes. Type 1 and type 3 requires ATP for the restriction activity, while type 2 doesn't require. So most of the well-known common restriction enzymes are the type 2, EcoR1, BAMH1H, HIN3, and the others like EcoK1, EcoA1 are type 1, while EcoP1, EcoP15-1 are type 3. Cloning vectors. So they are small molecules for convenient manipulation. They carry the target genes. They are capable of prolific replication in a living cell for amplification of the insert, the gene of interest. They must have convenient restriction sites for the insertion of the gene of interest. It would be beneficial if they have any marker genes for identification of recombinants. There are many types of cloning vectors. The most common ones are the plasmid, bacteriophage, and cosmid. Plasmids are smaller, circular. They are of bacterial origin. They have an insert size of around at a maximum of around 15 kb or 15,000 base pairs. They can be introduced in bacterial cells by transformation. They're generally used for introducing drug resistance. Bacteriophages are of phage or lambda phage origin. They are double. They can introduce double-stranded DNA. They are up to size of 50,000 K and insert. They can take insert around 15 kb or 30 kb. They can be introduced either in phage viruses, then followed to the bacteria, or directly to the bacteria. Central part of the phage genome is removed and insert is replaced, thereby allowing it a little more space for a larger insert size than that of plasmid. Cosmids, on the other hand, are the hybrids of phage and plasmids. The size of their own varies. They can carry insert around 35 kilobases to 45 kilobases, sorry, kilobase pairs, and can be introduced in lambda phage viruses and then followed to bacteria. Once entered into the bacterial cell, they replicate as the plasmids do. So now we will be showing stepwise simply how gene cloning is done using plasmid vectors. First of all, extraction of genetic material. Bacteria has two types of genetic material, chromosomal and extrachromosomal plasmids. Here we will be using a plasmid to express our gene of interest. Therefore, plasmid will be called as a cloning vector. Firstly, the gene of interest will be extracted from the cell. Now the dissection. Both the insert and the DNA plasmid are cut using restriction enzymes. The fragments are mixed together, allowing base pairing between their complementary sticky ends. Then DNA ligase is added, which covalently bonds the sugar phosphate backbones of the fragments whose sticky ends have already base paired. Insertion of the vector into the bacteria. The recombinant plasmid with the gene of interest is then inserted back to the bacteria. The bacteria is cultured on suitable selected media, which help us to differentiate colonies with plasmids without the ones which doesn't have. The gene of interest can be used for basic research purposes, also can be used to produce protein products. Thank you.